Hello students, today we will be learning about enzyme activity assay. Aim of the experiment: to determine the Km and Vmax of glucose oxidase using glucose as substrate. In introduction, enzymes are the catalyst of biological systems that determine the patterns of chemical transformations. They also mediate the transformation of one form of energy into another. The most striking characteristics of enzymes are their specificity and high rate of catalytic power. Enzyme kinetics deals with the quantitative measurement of the rate of enzyme catalyzed reactions and the systematic study of factors that affect these rates. Kinetic analysis provide the number and order of the individual steps by which enzymes transform substrate into products. Glucose oxidase is a cytosolic enzyme that catalyzes oxidation of beta-D-glucose into hydrogen peroxide and D-glucono-1,5-lactone, which is further hydrolyzed to gluconic acid. This enzyme is produced by certain species of fungi and insects and displays antibacterial activity when oxygen and glucose are present. Glucose oxidase is widely used for the determination of glucose in body fluids and in removing residual glucose used for gluconic acid production, removal of oxygen from food and other agricultural products. Commercially, glucose oxidase is also commonly used in biosensors to detect glucose. In this experiment, our aim is to calculate the enzyme kinetics parameters such as Km and Vmax of an enzyme, glucose oxidase, using glucose as substrate. In the reaction catalyzed by GOX, one molecule of glucose is converted into one each of hydrogen peroxide and D-glucanolactone. Then, the liberated hydrogen peroxide is reacted with a coloring reagent which consists of 2,2-bisinconinic acid and trichloroacetic acid. The cupric ion complex of 2,2-bisinconinic acid is reduced to cuprous ion, which is a colored complex. The absorbance of the formed complex is measured at 562 nanometer. The amount of product catalyzed by GOX is directly proportional with the optical density of the complex. Using Michaelis-Menten's equation and Lineweaver-Burk plot or reciprocal plot, the value of Km and Vmax are calculated. Using the formula, V naught is equal to Vmax into the concentration of substrate divided by Km plus the concentration of substrate, which is the Michaelis-Menten's equation, where V naught is rate of reaction, Vmax is maximum rate of reaction, and S represents substrate concentration. Km is Michaelis-Menten's constant. In materials required, we require 1 millimolar glucose oxidase, 2% glucose, 0.05 molar phosphate buffer, coloring reagent, 2% tartarate solution, bisinconinic acid and trichloroacetic acid, hydrogen peroxide, as well as copper sulfate. In procedure, in the first step, 2% potassium sodium tartarate and 4% bisinconinic acid was prepared in 0.05 molar phosphate buffer at pH 7. Also, 20 millimolar copper sulfate was prepared in water. The copper-containing color reagent is prepared by solubilizing 10 volumes of potassium sodium tartarate solution, 9 volumes of bisinconinic acid, and 1 volume of copper sulfate solution. The final concentration of 0.8% trichloroacetic acid solution prepared in 160 millimolar phosphate buffer of pH 7 was diluted in the solution mixture. The color reagent is prepared afresh each time before use. Next, a solution of fixed concentration of glucose oxidase at around 1 mg per ml and different concentrations from 40 to 200 micromole of glucose, which is substrate, were prepared in phosphate buffer of pH 7. Next, 0.1 ml of enzyme glucose oxidase enzyme was mixed with 0.9 ml of different substrate concentration, and the mixture is incubated for 30 minutes at 37 degrees Celsius. In the next step, after incubation, 2 ml of color reagent was added into the mixture. Next, the mixture was further kept for 30 minutes at 37 degrees Celsius for developing color. In the next step, after incubation time, the color development was ceased by adding 0.1 ml of 0.2 molar EDTA solution. Next, the absorbance of the solution was measured at 562 nanometer. A solution without enzyme was used as internal blank in all measurements. 3 replicate data were recorded from 3 vials containing same enzyme and substrate concentration. Next, a standard calibration curve was drawn using known concentration of hydrogen peroxide. In this step, the 2 ml of color reagent was mixed with different concentrations of 0.02 millimolar to 2 millimolar of hydrogen peroxide, and same procedure was followed as in test experiments. In the results, the standard curve of hydrogen peroxide provides the relative concentration at different absorption of the complexes formed. The straight line follows the following linear equation, which is y is equal to 13.767 multiplied by x. From the above equation, concentration of product, which is hydrogen peroxide, formed was calculated from the absorbance. From the above observations, the rates of reaction at specific concentration of substrate were calculated. Then, a graph of reciprocals of substrate concentration and rate was plotted to get Lineweaver-Burke plot. A straight line with an intercept at y-axis with the following equation was obtained. Here, y is equal to 16964 multiplied by x plus 18.72. The equation is compared with Lineweaver's equation, which is 1 by v is equal to product of Km by Vmax multiplied by 1 divided by concentration of the substrate plus 1 by Vmax. So, the intercept at y-axis is 1 by Vmax and slope of the line is Km divided by Vmax. Therefore, Vmax is equal to 0.0534 millimoles per minute and Km is equal to 906.19 millimoles. In conclusion, glucose oxidase is an enzyme which oxidizes glucose to gluconolactone, oxygen to hydrogen peroxide. Some fungi express glucose oxidase at their surface to produce peroxide which can kill bacteria. Using the properties of glucose oxidase, the blood sugar level can be monitored easily, conveniently, and economically. In the present experiment, enzyme kinetics parameters, which is Km and Vmax, were calculated using a fixed concentration of enzyme, that is 1 mg per ml, and different concentration of glucose substrate. The Km and Vmax of the glucose oxidase is found to be 906.19 millimoles and 0.0534 millimoles per minute respectively. Thank you.